So word. Start early? Fine, right? So we'll get out here earlier. So last week we talked about the first biochemical experiment about how scientists study cell cycle, and by looking at morphological changes, they could tell there's something in the cytoplasm that changing the stages of the cell cycle. Okay? If you were in my class last semester, you will know how to identify the nuclei by looking at the morphology of chromosomes. Right. Everybody forgot. Okay. So that's Rao and Johnson's cell fusion experiment. So scientists want to know what is or what are the factors influence the progression of the cell cycle. So when they look at the Xenopus oocyte, the Xenopus is that frog. Okay? And Xenopus oocyte look like the bubbles in your bubble tea, okay? When they observed the whole process, they found the process of oogenesis. Do you know what is oogenesis? The growth of your oocyte. Okay? So oo means oocyte. Genesis means development. Got it? So when they look at the process of oogenesis, the cell grow without splitting. So the cell get bigger and bigger. Just like this, get fatter and fatter, right? So that's grow without cell division. So when the oocyte become embryo, what happened? Fertilization, right? Then cells start to divide. However, they don't grow. Cell division without growth. Okay. So that's when it starts to divide, that's fertilization started. So they wanted to know the factor in the oogenesis and the factor when they start to do division. What's the differences? What makes the cell goes around the cell cycle? So yes, here's the Xenopus. We don't see these frog around campus anymore. Do you know Dr. Jiang? That was her the animal she was studying before the animal permission requirement. And she was trying to cut the nail of all these Xenopus around the campus, and then try to do the genetic tagging. Yeah. That was her, yeah. But she's not doing the frog anymore. It's a vertebrate. So during the oogenesis, you have the oocyte arrests at G2 and starts to do meiosis, and then arrests at meiosis 2. And after fertilization, you start to see the first cleavage of the embryo. So when you go back to your country and one day you want to become a teacher, I don't know about the regulation in your country. Do you need animal permission to do the animal experiment? You don't, right?
We do, but you still we do, but
It's not restricted. It's not
really restricted.
Not really.
Anybody can do the experiment.
Not anybody.
We have at least. I've opened a frog before in high school.
I did when I was 6th grade, but we can't do that in here anymore. For the law in this country, student under senior high, that's elementary school and junior high, should not have any animal experiment.
Which means
Should
we're
not.
not really strict.
We're not restricted.
Yeah, we're not really that strict.
Well, even yeah, in our
Any campus.
undergraduated, we already like
Using
having
a rat.
with
Maybe
rats.
junior high.
And also-
I opened a
Frog.
chicken.
Oh, yeah,
You
really.
open the chicken.
Opened a chicken.
You open the chicken.
I
But
don't think there's one.
Raw.
when I was 6th grade, we can open the frog. Yeah, but it was horrible because majority of the 6th grader, we didn't have training for animal dissecting. Yes, even though we were working as group, there are 6 of us. So there are 6 frogs in each group, right? And some of them didn't know how to start the dissection by just cutting the skin lightly. So when they cut try to open the stomach of the frog, they try to stab the animal, and then you see the blood just splashing out immediately. Yeah, not very pleasant class. It wasn't. Yeah. But one thing about the frog, you can do experiment without killing them, is to obtain their oocyte. And you can observe development of the oocyte or observe the development of the embryo in their nature environment. Try not to, do any experimental things on these embryos. And these embryos are very easy to make observations, since they do look like bubbles, right? It's huge. Have you seen the oocyte, the egg from mice or rats? Yeah.
No.
It's smaller. It's smaller than this. Yes. Have you seen humans?
Is it smaller?
Yes.
Oh my God.
Smaller than the mice or rats. Yes. Shocking, right? So, well, you think it should be the other way around, but the human's oocyte is really tiny. Require microscope to do a lot of manipulation. Yeah. Next time I'll show you the human's oocyte. Picture. So this scientist, Japanese, Masui, in 1971. I was born.
Lol.
No, I was born before that. Yes, he tried to set up an assay for identification and isolation of factor that cause cell division. Okay, so if the factor can cause cell division, if he inject the factor inside an immature oocyte, it will start to do division. Okay, it's very simple. When the cells start to do cell division, you will see the changes in the nuclei, especially when the nucleus goes to the metaphase, right? So that's the most important feature you should look for. And so what he did was taking cytoplasms from a mature oocyte. A mature oocyte is oocyte ready to go into being fertilized into an embryo, okay? So right before fertilization, it's a mature oocyte. If he took part of the cytoplasm and then inject back to immature oocyte, Immature oocytes are the oocytes that arise at G2 stage before meiosis 1. Okay, that's the immature one. And then he starts to observe what happened to the nuclei of this immature oocyte. He found that cytoplasmic factor from the mature oocyte will activate the immature oocyte going through the oogenesis. And he can amplify the stimulation effect by going through several rounds of this kind of like a backcross stimulation, taking the mature one back to the immature one. So he set up, Dr. Masui set up he called the factor from the mature oocyte cytoplasm maturation promoting factor. Promoting oocyte maturation. Simple as that. Okay? And he also had an assay to do the evaluation. So that's why he's important and he's famous for. So the Indian scientist is coming in. The Dr. Masui only tell you, set up the assay until there's a factor. He tried to find what is in the MPF. So he extract the cytoplasmic fraction from the oocyte, and then he tried to label the cell proliferation, well not cell proliferation, label the protein expression by incorporating radioactive methionine. So whenever the cells start to make a lot of new protein, that protein will have radioactivity on part of its structure. And then you can see it on SDS page to evaluate. All you need to do is to expose to an X-ray film. It's very easy, really fast. You don't need to do primary or secondary antibody in order to observe the film. Don't. So he found whenever you have the activity of MPF, something is here. And that protein is not there before the MPF activity. And it's not there after the MPF activity. So, interesting. Actually, the protein should be stable, right? But the protein, after synthesized, being degraded rapidly. And it looked like it's matching the peak of the MPF activity. Okay, that's his contribution. Okay, he found some protein. He doesn't know what is that. So let's summarize these two experiments. There is a factor, MPF, and the activity of the MPF can be detected during mitosis. And there are proteins that increase and decrease in terms of the MPF activity. And they call this oscillation phenomena is called a cycling. That's why it's called cell cycle. That's where cell cycle is coming from. Okay. It's the activity is cycling. The MPF, well, it was started as maturation promoting factor. Nowadays we can call it MPF mitosis promoting factor. It depends on your focus. Some people who study in cell cycle molecules, they will say it's a mitosis promoting factor. But if you are working with biochemical function of these molecules, you will call them maturation promoting factor. Okay. So either one of them is fine. The MPF activity is increased in mitosis or meiosis, and is decreased in the interphase. So Tim Hunt, he's the person who has the Nobel Prize. So Japanese and Indian scientists, they didn't get the Nobel Prize because they didn't answer the question, why, how, who, okay? So, Tim Hunt working on the sea urchin, and he did the same thing, just like the Indian guy, labeling the newly synthesized protein. And the way he evaluate cell cycling is by whether by looking at precise time frame with mitosis or interface. And he called these two protein most prominent protein that goes up in mitosis and goes down in interface, cycling A and B. Okay? That's the two most prominent protein he found that goes cycling with the MPF. But that's sea urchin. If you go to the beach and you want to do experiment for your student, you could go pick up some sea urchin, and then all you have to do is to inject high concentration of salt. Yeah, and all these embryos that will release.
Oh.
Yeah, you don't have to try to control your salt consumption. You don't have to kill the animal because you want to eat it, right?
Yeah.
And the oocyte or the embryo of the sea urchin is very transparent. It's even better animal model to study embryology in terms of if you study the morphology, looking at the microscope. So this is what Tim Hunt did. He also labeled newly synthesized protein with isotope. And he found A, B, and C. For most of the textbook, they didn't mention cycling C. Because there are so many proteins that goes cycling with the MPF. He only choose the two major ones. Okay. This is the cycling activity with the protein and the biochemical activity. So he did both of them to confirm the A, B, and C actually going cycling, and the activity, the level of the protein goes up and down, cycling with the same pattern of MPF. Okay. If you wanted to know how he did the experiment, you can look at this paper. Did I have it in there? I'll try. So, from Tim Hunt's experiment, he found there's a group of protein cycling. Okay, but MPF, does MPF equal cycling? The answer is no. Okay? The answer is no, because MPF have kinase activity. Why? How does Tim Hunt knows that? What does kinase do? Kinase. Phosphorylation. Okay? So if you do a kinase activity assay, you should see phosphorylation faint. But by incorporate cycling, you don't see the kinase activity. So they know there's something else inside the MPF. And they are not too sure whether cycling is part of the MPF or not by that time. So the answer, well, the problem or the question was answered by the genetics. Okay, and we're not going to go into there. Just jump up there. I will talk about the genetic part. Let's jump from there. After the genetic analysis, they know MPF is a complex of cyclin CDK. Okay. CDK is cyclin-dependent kinase. K is the short for the kinase. And the reason they know that is from the genetic study. Okay, so that's how they got the Nobel Prize. So cyclin is just a family of protein controlling the progression. Control. Okay? The way they control is by activating the kinase. And the kinase will phosphorylate its target gene to do whatever is supposed to be done. Okay? That's very important for MPF. The MPF can only activate during the mitosis. So someone needs to control the kinase. Cycling, the amount of cycling will determine whether the kinase activity is high or low. So something else, you need to tell the cycling goes away or comes back. If someone is doing the controlling. So the cycling was isolated from the sea urchin experiment. The CDK actually was discovered from the genetic experiment. We'll talk about that later because it's a genetic experiment from both East Saccharomyces and Saccharomyces bombay. Two different genetic analysis. So let's put these two together. One is cyclin, the other one is CDK, cyclin-dependent kinase. And this complex is MPF, no other stuff. So the function of this MPF is activation of its target protein by phosphorylation. So the CDK will phosphorylate the substrate. Okay, and activation of these target protein will cause everything you see in mitosis, all the morphology, including microtubule instability, including chromatin condensation. All these things you see, morphological change, are due to the effect of kinase activity. Okay, so phosphorylation of the GM130 will cause fragmentation of Golgi and ER membrane, and therefore this protein has a phosphorylation site that would affected by the kinase. Got it? Okay. So all of them could be a target for the CDK. So the cyclin, there are so many cyclin and so many CDK, and these information are coming from the genetic study. These CDK are serine surrounding kinase, meaning they phosphorylate a particular residue, serine residue and surrounding residue, okay, of the target protein. And they are constitutively expressed, meaning the concentration of this protein is the same throughout the cell cycle. Unlike the cycling goes up and down cycling, okay? So the concentration of CDK is always the same. It's always the same. How does the CDK gets activated? It's there all the time. So when cycling shows up, well, cycling is the one gets control, the concentration of cycling. And we will talk about how it got control. Before that, let's look at how it controls the CDK. When cycling binds to the CDK, this complex actually is an inactive form. Okay, it needs to be phosphorylate twice, and there are 3 phosphorylation sites on this CDK. Okay, 2 of them are inhibitory inhibitory site, and one of them is the activation site. And you need third enzyme, also you have 2 kinase to do phosphorylation at 3 sites. And you need a phosphatase to remove the inhibitory phosphate in order to activate the kinase. Okay? It's really a little bit complicated, but let's take a look like this. So this is your cycling, and this is your CDK. There are 3 phosphorylation sites on the CDK. One is the activation one, the other 2 are the inhibitory one. So when cycling binds to the CDK, it's an inactive form. On the top of this complex, there is a CKI, meaning CDK inhibitor, is there before cycling comes in. Okay, it's there before cycling comes in. That's a CDK inhibitor. What happened is you need We1. We is a kinase that will phosphorylate those two serine surrounding site on CDK. These are the inhibitory site, meaning if you have phosphorylation on here, inhibit kinase activity. That's why it's in black. The one on the right-hand side, that's an activation site. But this activation site is blocked by CKI. So when you phosphorylate by the we, you change the conformation. The CKI will no longer binding to the complex. Okay? Oh, so you need the phosphorylation to remove the CKI. Now you expose the phosphorylation site of the active one. The CDK activating kinase will come to phosphorylate this active site. Got it? So there's a sequence, and there's someone comes first, and then the second one, and the third kinase comes in. So that's CKI. It's there when cycling come to bind to the CDK, and We1 will come to phosphorylate the inhibitory site. We1 is not capable phosphorylate the activation site because it was blocked by the CKI. Right. So when We1 phosphorylation, after We1 phosphorylation, the conformation change, the CKI will leave. And now you expose the phosphorylation site for CAK to do the phosphorylation. Now the problem is the inhibitory site has phosphorylation. We need to take it away. So we need to ask a phosphatase, CDC25. CDC25 is a phosphatase. Okay, so anything take away the phosphate is a phosphatase. It's dephosphorylation. Okay? And anything give the phosphate group is kinase. It's called phosphorylation. Got it? Okay. So that's sometimes a little bit confusing. So you need CDC25, a phosphatase, to take away those 2 inhibitory phosphate in order to activate this cycling and CDK complex. Now your CDK can phosphorylate more CDC25 to activate more CDK. Okay? So you could go phosphorylate. Well, the CDK will also phosphorylate another molecule. We'll talk about this molecule later. Okay? It's called APC. APC is a molecule in ubiquination system. Do you know the ubiquination? I will talk about it later. And what this ubiquination does is to destroy ubiquinated protein. Okay, so you will add ubiquitination on cycling. So at the same time, when you activate the CDK kinase, the kinase will phosphorylate APC. The APC will ubiquitinate the cycling. The cycling will be destroyed. That's why you see up and down of the cycling concentration during the cell cycle. Okay, so somebody must regulate this APC, right? Okay. So in summary, the activation of CDK involves phosphorylation and dephosphorylation. There are two inhibitory sites will be phosphorylated first, and then removal of CKI will expose the active site for CAK to do the phosphorylation. That's CAK. The final step is requiring the phosphatase CDC25 to dephosphorylate the inhibitory site in order to fully activate your CDK. Okay, so that's just a summary. If you kind of confused by my explanation, you can look at the Campbell textbook. This one, this is coming from Campbell Pierce. Yeah. So CDK, there are so many CDK in the mammalian cell. Okay, and for each time frame in the cell cycle, they have different CDK or different cycling to do the work or to do the job. For mitosis, you know there are cycling A and B, right? Goes up and then disappears. So mitosis, the CDK for the mitotic phase is CDK1. For the interphase, what is the longest phase in the interphase?
It's a G1.
G1 phase, okay? So most of the time when we were studying the cancer cell, when we want to know whether the cell go into the interface, we will look at the CDK and its partner, the cycling, the CDK4's partner, okay? And so for the S phase, the CDK is CDK2. For the G1 phase, CDK4. Sometimes it's CDK4 or sometimes with a minor population of CDK6. So in most of the paper you read, they will look at CDK4 and 6 together. So you should have an antibody that could detect both of them at the same time. So for the cycling, the G1 cycling is cycling D, and the mitotic cycling is A and B. S phase will have E and A. So you have A and S phase and mitotic phase. Why? It's gradually increasing at S phase. So you can detect cycling E and A at S phase. So if we look at, well, that's how you partner CDK and cycling together. You don't have to memorize it, but you need to know CDK4 and 6 should be looking at in the G1 phase, and their partner is the cycling D, D1, 2, or 3. So they are isotype or D founded in the cycling D. And for S phase, usually is CDK2 and A. In the mitotic phase, we're looking at CDK1 and cycling A and B. So that's the most important one you should remember. So if we draw the time frame like this, you have G1S, G2N, and that's another cycle, okay? Your CDK4 started from end of your mitosis and go into your G1 phase. That's why you have CDK4 and CDK2 is right here. This is CDK1. Sometimes you have CDK6 right here, okay? And then for cycling D gradually increase at the beginning of G1. So it's a little bit different like CDK. And then your E has overlapping with D, and A has overlapping with E, and B has overlapping with A. Okay. So why the cycling has this up and down situation? So the protein expression, and somebody killed it. So the amount of protein drops rapidly. So something needs to go away in order for the cell cycle to proceed, okay? Because the target for each CDK is different. At different stages of your cell cycle, you need to activate different proteins. So cycling is trying to control when does this kinase should be activated. So CDK1 should activate all those proteins required for cell growth G1 phase and then go into the S phase. Okay? But the requirement is no longer there when the cell is in the S phase. So we need different cycling to activate different kinase to do the work, to activate protein required for S phase. Got it? Okay. So ubiquitin. Ubiquitin is the killer that's going to kill all the protein that is not necessary to be there. Okay? And that's one of most efficient way to control the existence of the protein. If your protein can generate the new protein by itself, some of the protein you cannot generate rapidly, but for the cell cycle, it's fine. Ubiquitin is a protein degradation system. For the targeting protein that will be destroyed by ubiquitin, this target protein should have a destructive box. Okay? That is the region the enzyme will hook up the small molecule on the destruction box. And that gives the sign saying that this is the protein to be killed. Okay? So for example, this is a cycling. The cycling has a destruction box at the end terminal. And some of the destruction box kind of has a conserved region. You don't have to memorize it. All you need to know is the destruction box for ubiquitin is conserved. There's a conserved region, conserved sequence for it. And that's where you have enzyme to bring ubiquitin. It's a molecule. Ubiquitin is a molecule, small tiny molecule will hook up on the box. Okay? And now become a target for killing it. Who kills the protein? The ubiquitin won't do anything.
Enzyme?
A organelle. Well, not really an organelle. A complex.
Proteasome?
Proteasome, yes.
Proteasome.
Proteasome is it's a very interesting. Or is it an organelle? I don't know. So here's your APC.
It eats everything in a way, right?
Well, it's not eating. Proteasome is a complex of a lot of proteins assembled together, and it's assembling like a polar structure. They have ATPase facing the lumen of the structure. So the protein that to be protease sending to the proteasome or passing through the tunnel, and then you be the protein over the polypeptide will be well hydrolysate without specific site, just being cut randomly when they're passing through the lumen. So you have tons of the ATPase that line up on the proteasome. It's a structure look like, this proteasome has a structure looking like chaperone. Do you know what's chaperone?
The one that initiates.
Chaperone? Hisha protein is a chaperone? Do you know what is chaperone?
The one that initiates transcription.
No. Chaperone, do you know what is Hisha protein? Hisha protein. When the cell gets stressed, Hisha protein. Okay? When you increase the temperature for E. coli from 37 to 42 degrees, the number one protein that has great expression is the Hisha protein family. Got it? All the Hisha protein are ATPase, meaning they bind to ATP to do their work. What did they do during the heat shock? What did they do? What did they do? Oh, you need great expression of these proteins. Why do you need a lot of these proteins for during a heat shock? What happened during a heat shock? Heat shock. What happened when you
Protein degrades.
boil your milk?
It's like degrade.
What happened to your protein?
That's stress.
Aggregate.
Aggregate. Yeah.
Yes, it's not degradation. No, it's not
Fold.
degrading. It's misfolding, or it's in a structure that is not in its native way. Okay? Denaturation. Sometimes it is aggregated into clumps. Okay? So the role of the heat shock protein is when the cell is under the stress or heat shock, it will come out and binds to non-specifically binds to all these proteins by preventing folding, misfolding, or becoming an aggregate. Okay, so that's one of the function of the chaperone. What is the second function? So HSP70 is the major heat shock protein in your cytosol. Okay. Under the heat shock condition, you will increase the amount of heat shock protein maybe by 10-fold, I don't know, 7 to 10-fold. But the amount of the HSP70 under normal condition it still occupies 10 to 20% of your total protein. 10 to 20% of your total protein is a lot. Why does a cell need a lot of heat shock protein without stress? Why?
So they can withstand the heat.
No, under the normal condition, it's cool. It's in an AC condition. In normal, you still need chaperone. That's why it's called chaperone.
So the protein can acclimate to temperature
No.
according
Temperature is fine. No temperature factor. Think about it. Why does cell require chaperone?
So it doesn't like misfold when you wait.
Yes. When?
As it splits, so it doesn't. instead of like a mutate, so that
No,
because
we're talking about normal, not
normal
mutate.
condition.
No, normal condition. We know
It needs
under
a proper-
stress, it comes out a lot.
It just needs a proper structure for the protein to function. And
Proper structure.
so basically
Control.
So it has two functions. It's really important. One is when you are when the gene is doing the transcription and translation,
right?
right? So when it's translating, what happened to the protein that is not going to the ER? If the protein is going to the ER, meaning it's going to the secretory pathway, right? So going to the ER, going to the Golgi, and excreted out to outside the cell. If the protein is going to mitochondria, does it need to go to the ER and Golgi? No. Or if the protein is going to stay in the cytoplasm, does it need to go to the ER? No, right? So what happened to those proteins that is not going to the secretory pathway when it's translating to the cytoplasm?
Fold randomly.
Exactly. It is folding. You need to fold. The polypeptide does not do anything at all. You need to fold into a proper structure in order to function, right?
Right.
So if you fold too fast, it becomes aggregate. It's useless. It's threatening the cell. You need somebody to slow down the folding. So you need to bind to the chaperone.
Oh.
So the role of the chaperone is to be there to
Make sure the protein folded properly.
exactly. That's the same function of the Hishap protein inside the ER. So majority of the protein folding QA and QC is happening in the ER because you have glycosylation and you have ER Hishap HSP70 that is assisting the folding properly. Misfolding will stay in the ER and become UPR reaction. Have you heard UPR? Unfolding protein response. Unfolding protein response. Okay? Go back and look at your cell biology. So the second function of the Hishap protein is try to maintain translocation competence. If it's not staying in the cytoplasm, it requires to go to lysosome, or it requires to go to mitochondria. And the mitochondria has particular translocator that will not allow a huge, humongous protein to go in there. So all the protein that goes into the mitochondria should be in a translocation competent form, like a polypeptide form, to go into the mitochondria. Okay, that's the second function of the chaperone. So the definition of the chaperone is to bind and assist folding of all the polypeptides. And itself will not become part of the final structure. That's very important. Okay, sometimes when you are binding to a polypeptide, by mistake it becomes part of the subunit or part of the protein. But the chaperone comes in and goes out. And that's why it requires ATP hydrolysis as an energy source to leave the polypeptide. Okay? Binding could be random, could be spontaneous, but leaving requires some energy. Okay? Inside the ER lumen or inside the Golgi, you have particular set of your Hishap protein, especially those in the ER. When the polypeptide goes in from the ribosome, okay? The Hishap protein also binds to the polypeptide. Number one, prohibit misfolding. Number two, try to pull the polypeptide into the ER lumen. So it has additional function for the heat shock protein in the ER lumen. I'm talking about ubiquitination. Could be at one side, ubiquitination could be at several spots of the polypeptide. Okay, these are just different mode of ubiquitination depending on whether the polypeptide bearing the conserved sequence for ubiquitination. So we know any protein has ubiquitin on it will be sent into a ribosome, not ribosome, proteasome for degradation. The protein will be degraded into fragment randomly. Okay. So these ubiquitin will be recycled, can be reused. Okay. So you don't need the cell to ask the cell to express or to make a lot of ubiquitin. They could be recycled after the proteasome kill all those protein. The ubiquitin will bind to E1 enzyme. So E1, for every system, they have their own ubiquitin system recognize the ubiquitin. And we will talk about our ABC later. And so E1 is the enzyme that binds to ubiquitin. The E1 will, and look at the substrate. The protein that receives the ubiquitin is getting the ubiquitin from E2. Okay, from E2. So the ubiquitin should be passed from E1 to E2, right? The reason that E2 knows to give ubiquitin to this protein is because E2 recognizes E3. So by binding to E3, it transfers ubiquitin from here to there. Okay. So E3 does not bind to ubiquitin. Does not. E3 does not. It recognizes the protein target. So E2 is the enzyme that's receiving the ubiquitin from E1. So you have E1 binding to ubiquitin, bring the ubiquitin to E2. E2 will recognize E3. The E3 will recognize the target protein. And the ubiquitin will be transferred from E2 to the target protein. Okay? Got it? A little bit complicated.
Yes.
So how do you analyze on the SDS page? Okay, you have to know whether your protein is ubiquitinate or not. So these three are immunoblocked, meaning result from Western blot. Okay, so that's the antibody for the Western blot. So this is the protein. The NIEL is a protein. When in the wild type, the size of protein is 97 kilodalton, but at different strain, you will see various bands. If I don't do any experiment, just by looking at the first panel, I would say that's glycosylation, right? Because I'm just guessing. Because anything that is larger than the native form of the protein could be the modification to the protein. And the best guessing is glycosylation, okay? Because the molecular weight is really big. So in order to rule out the possibility of glycosylation, you can do a Western blot using antibody against ubiquitin.
Any kind?
Any kind. There's only one kind of ubiquitin.
Right.
Only one kind of
But if you
ubiquitin.
use any kind of...
You have a different kind of E1, E2, and E3. There is only one kind of ubiquitin. Okay? So the antibody for ubiquitin is cheap. Antibody for your E1, E2, or E3, they are expensive. Okay? Got it? So this is 97 kilodalton. And you will find that's ubiquination. Okay, it's not glycosylated. There are also different ways to differentiate whether these multiple bands coming from ubiquination or glycosylation. Another way to rule out possibility of glycosylation is to add glycosylation inhibitor. N-link or O-link. If you have N-link glycosylation, meaning is glycosylated in ER lumen. If you have O-link glycosylation, meaning that's your biochemistry. Go back and study your Zubei. O-link is in the Golgi compartment, and there are specific inhibitors for you to inhibit the glycosylation. So if you're adding the inhibitor and the band is still there, meaning it's not from the glycosylation. Okay? What is the biochemistry textbook you guys have? Which one? You don't want to.
It's not for biochemistry.
You don't have biochemistry?
Yeah,
We don't have the textbook.
there are no textbooks.
Do you have to take biochemistry this class?
I mean, we did.
Yeah, but no textbook.
Yeah. We only need to listen.
Listen to
Say
me.
no. No, I mean like the wonder of space and unknown. We just need to imagine.
Holly, what is the textbook we use in here for the biochemistry?
Actually, I only read a PowerPoint.
I didn't read it.
Yes.
Oh, okay.
Instant study material.
You can look for the second-hand book, textbook, Zubei. Z-U-B-Y, Zubei.
Super.
Yeah, that's a very good biochemistry textbook. If you can buy a second-hand one, buy it. It's a good investment. Or you can look at the old version of this biochemistry textbook in NCBI's website. Okay?
John Zube.
Zubei, biochemistry.
Yeah, I found it.
You found it, right?
Zubei?
Yeah, that's the one. My dog loves it. He chews on it all the time. Zubei.
This book looks depressing.
It's not depressing. Okay, so let's look at APC. Remember I said APC activation of APC will cause ubiquination of your cycling, right? Okay, so APC is a short for anaphase promoting complex. Okay, so this little molecule comes activated during anaphase. That's where its name coming from. It regulates the activity of cycling. So when it comes out, it will give ubiquitin to cycling, and the cycling will go to the proteasome. Okay, so that's the missing link of why these cycling are doing the cycling. So if this is the cell cycle in a different way, so now the interface is right here. Okay, that's the interface. All the red colors are the M phase. This is the prophase, metaphase, anaphase, and theta phase. Okay, so that's our M phase. This is the MPF. So we have the cyclin B and the CDK. CDK 1? Okay, so at metaphase we have high cyclin concentration and high MPF activity. Okay, so that we have cyclin and CDK. So this CDK, the kinase activity during the metaphase will also phosphorylate APC. Okay. So when the activity of MPF is peak at the metaphase, this activity phosphorylates all its target. One of the target is the APC. When the APC is phosphorylated, it becomes activate. Activation of APC will binds to cyclin. Okay, so by binding to cyclin, it gives those ubiquitin to the cyclin from E2. So the ubiquination is happening in anaphase. Ubiquination is happening in anaphase. So at anaphase, you start to see drop. The concentration started to decrease, and then at the end of the mitosis, they should be all gone. All the cyclin should be gone. Okay. So that's the APC become inactive. So there is another molecule, is a phosphatase, come to dephosphorylate the APC. Okay, so there are some other regulatory protein here to inactive your APC, and then allow the cell can go into the interface or go out of the cell cycle. So the cyclin drop at the late anaphase, and then you start from G1 phase. So that's M phase, G1S, G2N. APC started work in the metaphase and anaphase. Get phosphorylated in metaphase and polyubiquination at anaphase. Another molecule doing the same thing is at S phase. It's called STMB. Okay? You don't have to memorize it, but you have to know there's always some molecules come to apart, okay, in the cycle, right? So SPF is doing the same thing. It's CDC28. And
CDC?
Yeah.
It's the same.
No, it's different CDC. The other one is CDC
CDC 20.
no, APC is not. APC is called APC. Doesn't have the CDC on it. Let me see. Yeah, it's just APC. It's CDC 20. 28 is for SPC, okay? SPF. You don't have to memorize it. Just have the same system is happening in S phase. Okay. Now look at the missing part. It's the genetic study. If you like all those e-study, you can look at the Campbell's chapter 12 or molecular biology by Lodish, chapter 13. You can find the electronic version of the Lodish textbooks in the NCBI, but you need to search using the keyword. Okay, for example, Hartwell or Polners. Okay, so Hartwell is the scientist who used Saccharomyces cerevisiae to study the cell cycle, the budding yeast, the yeast from the beer. No, beer. Yeah, not wine. And Polners using Saccharomyces bombay. So that's Saccharomyces bombay. That's Saccharomyces cerevisiae. They look different, but both of them are good for cell cycle study, since you can identify the morphological changes under microscope. Have you observed yeast under microscope in Jackie's lab? Yeah? Look at their cell cycle, okay?
Yes.
So they both started the experiment in their own lab, and they're kind of like enemies of each other. I don't see both of them appear in the same conference. Never. Because
Why?
I don't know. I was in the East Society group in New York, and every time we see the Howard will show up. Toners never show up. And he belongs to another East group. But the East Society is very, it's not smart. They are the first community that gather together to sequence the whole genome of the organism. So yeah, sometimes they.
How much money did they spend?
Well, Harvard is in the Cold Spring Harbor. Cold Spring Harbor has a lot of money. Don't
Oh.
worry. And Polners is in Yale. It's either in Yale or it's in Rockefeller University. It's
Okay.
really rich too. Yeah.
Insane.
When I was in University of Washington, every lab can, if you don't have any grant, because you failed grant application this year, but you can still do your experiment since they have a central supply office that will give you minimum support of all the experiment you needed. Yeah. I think there's an allowance for minimum study you can do. And the same thing in China Medical University here. Yeah. You don't need to spend too much time writing grants. If you, they prefer you spend your time submitting paper,
Okay.
not writing grant. Okay. They think writing grant is wasting of time.
Understandable.
Yeah. So in China Medical University, yeah, Taichung, yeah, that's the one in Taichung. It's a private school, that's why they can do that.
Oh.
Each lab have $50,000 to do every month
What?
to do the research without any grant. Wow. Yeah. And
Shit.
if you have, well, you don't get really good pay though. For a postdoc, I
much?
think the minimum is somewhere around 50,000 to start with
Okay.
per month.
Per month?
Yes, per
Okay.
month. It's pretty low in here. Postdoc,
Oh,
not master. Okay, postdoc. You have a PhD.
Uh-huh.
You get 50,000 per month.
okay.
That's kind of low.
For a PhD, I guess. But I don't know. I feel so broke
But-
right now.
With that.
I'm sorry.
If you have a paper published, for example, you have a paper published in Nature series,
Mhm.
Nature Communication, Nature Medicine, Nature blah blah blah. You don't need to be first author. You don't need to be the communication author. You can get at least 300.
What the fuck? You serious?
Yeah, 300,000.
That is
300,000. Right?
yes,
300,000
300K.
per paper. The first author.
300K.
The first author for that paper, he got
Oh my god.
it's kind of different culture. From comparing public school and private school.
Yeah, it's always
yeah.
something different.
That's how they encourage you to write papers. I come from that
I feel
private
broke.
school, so I know.
No, that's insane.
So
Feels so-
you need to write a lot of paper
Uh-huh.
in order to get a lot of money. And you get cash for it. Cash. 2000. Yeah. That was like 20 years ago.
I started years ago, okay.
Yes.
What about them?
It's more than that. So, well, but they are willing to give you a lot of bonus, but it's a high stress working environment because you don't have anything, any production, meaning no paper. They're going to keep you for a couple of years and they'll say, well, we need to talk. And then next day you will be disappeared. So the way they do the sales cycle study is they want to find a mutant using mutation to study what happened in the sales cycle, which gene is required for the sales cycle to proceed in the mutant. Okay, so they have to know each stage in the sales cycle. Does the cell have different shape they can distinguish during the sales cycle? And can they grow as a haploid instead of diploid? It's easier for them to do manipulation when you are doing the genetic study. The problem is you need to screen the cell that doesn't grow, right? That's the mutant. Okay, so you have one strategy for any mutation is temperature sensitive mutant and then do the replica plating. Another strategy is using random mutagenesis by UV irradiation or X-ray irradiation or giving some chemical. But you have all the population that is mutated. There's no way to study its normal form. By using temperature sensitive mutant, the mutated form coming out when temperature is high and it comes back normally when it's at 37. Okay? Yes, yes. Okay, so you add mutagen at 20, well, since it's a yeast, the best temperature to grow, what is the best temperature to grow yeast?
37.
No, no,
28.
yes,
22.
under 25.
Oh,
Yes, under 25. If you, well, if you grow your yeast in 37 degree, you won't die, but
like
the
Slow,
protein,
slow, like slow.
no, it smells funny.
Stop.
Yeah, it's kind of like huge morphological changes.
Oh,
All the
wow.
metabolism is different, and it grows. Some of them will die before it reaches 37. Once it reaches 32 degrees, the smelling is not so pleasant. Okay, and the best temperature to grow yeast is somewhere around 16 to 17. It grows slow, but the yeast is happy in that condition. 23 is also good. Okay, it gives you fast growing and it gives you protein or metabolism that is normal. Okay. 23 is easier to control in a laboratory. If you wanted to grow something that is 60, you need a refrigerator, right? You need to control the temperature. Yeah, it costs more money. That's why it's 23 in here. Otherwise, they should use 16 or 17 degrees. Okay? And you add mutagen. Any mutagen can be added in this condition and allow the cell to grow for like 5 hours. What is the doubling time for your yeast? What is the doubling time for your yeast? Why is 5 hours?
It's fast to grow.
Why you need to play on the east outer plate. Why?
It's
After adding a mutagen, what happened if the east replicate twice or 3 times? So you will have mutant strain or 2 or 3 more colony here. It's really difficult for you to do the analysis. So you need to minimize complication by restrict the yeast culture under just one cell cycle. Okay, just one cell cycle. And place, how many plates do you need to plate? You need to find out the number of the gene that will appear in, or if it's a low copy number of the gene, you need to plate more times. You have to do the calculation. You need to grow all the cell cultures on the plate. For example, in order for you to be able to identify, each plate could have only 200 colonies. Does that represent all the gene? No, right? The yeast has more than that. So you have to count how many genes you have in the genome. And then, you have to screen maybe 10 or 20 plates. It's a huge job. Okay, and this is not a small plate. It's a huge plate like this, like a 10 centimeter plate. Then you do the replica. This is called replica plating. That means when you allow the cell to grow as a colony, that represents coming from one cell, right? After has colony grow, you make like a stem. Make a stem. And then at the same time, you could have one grow at 23 degrees, the other one grow at 37 degrees. And that's temperature, that's heat shock. If you have temperature sensitive mutant, it won't grow at 37. It will grow at 23 because it's temperature sensitive. Okay? So you can identify what happened to this colony from here, not from here. Nothing grow here. Got it?
What about if after you stroke the yeast is grown, but it's slow, so it's not sensitive in the mutant?
If you go too fast,
Like if it's grow in the 36, so it's not.
it still grows. It's not temperature sensitive. It doesn't have mutation here. Because if you have mutation, meaning you cannot proceed the cell cycle, you shouldn't have any growth. Well, it does have mutation, but it's not on cell cycle. Let's put it this way. Okay? Okay, so let's look at how Polners do the screening. So Polners using Saccharomyces Bombay, and it looks like a rod shape. So under the this is a cartoon. Cartoon represents a figure of this Saccharomyces Bombay on the G1 phase, S phase, G2 phase, and mitotic phase. Okay. So what happened in the mitotic phase? When the cell is ready to separate, you see what is this? Furrow.
What?
We talked about this last time, right?
Yeah. Oh yeah, we do.
Last week, go back and check your notebook. And cleavage furrow. Remember, it's a furrow. Some condensate, some U-shaped morphology, and that's the sign for mitosis. When it's in the G2 phase, the cell gets elongated and huge. That's what happened in the Bombay. So if in a wild type, For example, CDC2 is normal. During the mitotic phase, you should have a cleavage furrow like this. If you have CDC2 that is defective, the cell will not go into the mitosis. It gets stuck in the G2 phase. What did I say in the G2 phase? The cell gets elongated. The cell gets elongated. What happens when you see the cell smaller? And it does have the cleavage furrow, meaning CDC, D meaning gaining the ability of going through a cell cycle too fast. So it doesn't have time to grow, just like your cancer cell. Okay? That look like your cancer cell. Okay? Gain of function. That's loss of function, that's gain of function. So under the microscope, that's the wild type. That's the mutant.
They're much smaller.
Yes.
Yeah, they don't.
And that's CDC2 defective, elongated. And that's the wild type. Okay? So I should have another. By studying the morphology, he identified mutant V1 and CDC13 and CDC25, and he found that CDC2 is important for the cell to go into the M phase. That's the major one. And he also, that's this one, he also found that CDC13 is a CDK. That means when you have CDC13 is mutated, you don't have MPF, so you'll get no longer. Yes, you'll get longer. And then when he look at CDC25, when you don't have CDC25, you also get longer phenotype. If you have too much V1, it also has a long phenotype. So by gaining the function or losing the function, they get the same phenotype. Okay? Gaining CDC22, losing V1, you get the same phenotype. Okay? So by mixing and matching all these different mutants, they found that CDC2 is the major one from G2 to N. And then V1 and CDC25, one is actually stimulating, the other one is inhibit the CDC2. Okay? So another, so that on the one on the right is the Bombay. The one on the left is the Saccharomyces cerevisiae, the budding yeast. So by looking at the phenotype, that's probably interphase. That's probably S phase, G2 and M phase, probably, okay? And all the mutant strain that isolate from each genetic study has the name CDC. From Horner's Saccharomyces Bombay experiment, you will have SP in the front. For Saccharomyces cerevisiae experiment by Hartwell, you will have SC in the front because they name their own strain, and that confuses us all the time, right? Since they don't collaborate. So for Baker's yeast, the budding yeast, CDC is CDC28. It's the same strain that Pollner's identified in Bombay CDC2. Yes, that's the same. But take a good note that in Hartwell, he capitalized all the genes' name. In Pollner's experiment, all the genes' names is in small letter. Yeah, it's kind of chaotic when you are reading all these classical paper back in 1978 or 80s or 90s.
Paper?
Yes, it's in the paper, and they're talking about the same molecule, and they were discussing the same mechanism, but they're using a different name for the gene, and really confuse us. And that's why in 1980s they started with Bombay or Cilicia, the short in front of the gene. They don't want it to organize the genes name together. Not until somebody did the human's gene. Okay, so that's in the East. So Hartwell, this is G1S, G2M. The morphology for this budding is this G1 is the smallest one, and then budding, when you start to see the bud coming out, that's entering of the S phase. So sometimes the S phase phenotype look alike the M phase phenotype. The only way to tell the differences is to see there are two spots. One is two nuclei, okay, and that's one nuclei, right? Otherwise, well, the phenotype look alike because there's a bud. So identify the number of nuclei. Sometimes it's difficult to identify nuclei under the microscope. So look at the size of the neck where the bud is coming out. If it's small and narrow, it's probably in the S phase. And it's huge and big and huge neck. And that's the mitotic phase, okay? That's how you identify it. But it take practice. It takes practice to identify these stuff. Hartwell did something much more fun than Pollner's. So in a low temperature, he has all these different cells grow at different stages, right? So he started at the restricted temperature. He stopped the cell cycle at 37 degrees. Okay, so everyone started at G1. So he want everyone started at G1, and then grow at 37 degrees here. So just remember that diagram and look at the replicate plating. That's the 27 degree, that's the 36 degree yeast culture. So the one I circled, do you think they are mutant form or the wild type? Look at this one here at 23. Look at this one at 36. So is there a cell cycle going? Yes. Well, remember? 23 and then restricted temperature. So everyone started at the same time and we raised the temperature. So this one has cell cycle, right? From 2 to 4. The one above this one, yeah, has mutations, cell cycle arrest. Okay, so Lee Hartwell used different strategies to screen the temperature sensitivity strength. Either one of them is fine. Okay, either one of them is fine. And this way you can tell right away which one is the strain you're going to analyze. So you want to analyze this mutant, right? So this one is normal. This one is normal, and this one, it looked like mutant, but I couldn't tell. I really couldn't tell.
It's not normal.
That's normal. The one in green is normal. But that's another mutated. That's normal. That's mutated. Okay? So how do you clone the gene? Now we know we can identify the mutated clone, right? So you could, this clone is mutated. What he did is he used complementation assay. So what happened in the mutated form? The cell cycle stopped, right, at a high temperature. So he used cDNA library randomly, and then clone into a expression vector and send all these, every single plasmid into this mutated E strain to see which gene will rescue this mutation. Okay, so he doesn't know what is the sequence of the DNA in here. He doesn't know. All he knows is that he's putting the gene from the yeast library to see whether it will grow. If it grows, he comes back to purify the plasmid and sequence and give a name. For Lee Howell, all the mutation that allows has function in cell cycle, he will give CDC 1, 2, 3, 4, 5. So if you see any genes name CDC something, has to do with cell cycle, okay? And the number meaning is coming from the mutation analysis, just the order of the analysis. And then Polners, he read Lee Howell's experiment, and he really liked it. He thinks he's really smart, so he stole his idea.
Really stole.
He did. He stole his idea by studying the human gene. So if it works in the body yeast, or does it work in the human gene? Okay,
Cool.
so he used the same strategy using human cDNA library and working on the cell line. And he has to rescue the yeast, well, not cell line. He used human gene to rescue his base mutated form. Okay, and he has his postdoc. That's his postdoc. Did the experiment, and the human gene can rescue yeast mutation. Okay, yeah, that's his postdoc. I actually saw her.
Oh, really?
That's why I put her pic.
Cool.
Such a typical New Yorker.
A New Yorker.
Back in the day,
New Yorker street like New Yorker.
New York in the 80s.
Well, they have the largest yeast society in New York. Yeah. If you guys want to do postdoc, I recommend go to New York. Has a lot of resources, a lot of teaching opportunity. Yeah.
New York.
Yeah. Well, when we were,
I think anything is everywhere in New York. Anything exists in New York.
yeah, when we were in grad school, they need not teaching assistant, they need professor to teach anatomy in CUNY, City of New York. College of New York, sorry, College of New York.
College.
Yeah. And they have program for health-related professions, like
Psychologist?
yes, yeah, or rehab program, and they do need professor or teacher to teach anatomy. they were paying us like 30 years ago, 20 something years ago, $25 an hour. And the basic wage back then was $5 an hour. And they were willing to pay 25 to $30. The doctor gets paid for $25 per visiting. Emergency doctor gets paid $60 per hour back then. Yeah, so 20 something is a lot of money. That's a lot of money. Let's take a quick review and we will wrap up for today's lecture. We don't have time to talk about apoptosis today. We'll talk about it next week, okay? So we study in the cell cycle, we have G1S, G2N. And by studying the biochemical analysis, we know the MPF. If we look at the control, the regulatory point, the point that regulates the cell cycle. For example, if you have, the cell needs to make sure that everything is ready, the replication is finished and everything is replicated 100% correct, they will proceed to the G2 phase and then go to the meta phase, the mitosis phase. If you have DNA damage, it could inhibit the cells of S phase or G1 phase or G2 phase. Okay, so a lesion in the DNA damage is affecting many regulatory phases, not just one place. Remember this. But for each time frame in the cell cycle, you have a regulation gate to make sure cell cycle is going properly. Okay. So that's the review for your CDC, your cycling and CDK. So I'm not going to say too much about that. And there are partners for CDK in your cycling. The major one for M phase is CDK1 and cycling B. For the G1 is CDK4/6 and cycling D. That's the one you have to memorize. Okay, these two. For S phase, CDK2 and E. And that's CDK2 and A, CDK1 and A. So when the cell goes out the cell cycle in G0 phase, what happens? It stays there, right?
Stays there.
Doing nothing. When the cell receives a growth factor stimulation or mitogen stimulation, a mitogen, something stimulates mitosis. Okay. Growth factor, the cell needs to grow. It will stimulate the cell going from G0 into the cell cycle. Okay, so that's the signal. Any cell in the G0, when they wanted to go back to the cell cycle, they have to receive the growth factor stimulation or mitogen stimulation. Let's either going through G protein coupled receptor, just show me 12 o'clock today, or growth factor binds to the growth factor receptor. Usually it's a tyrosine kinase receptor. What leads to the binding of the factor to the receptor? Signal transduction pathway, okay? We'll talk about the signal transduction pathway. There are three major signal transduction pathways for any cell proliferation. Could be PI3K pathway or AKT pathway or ERK pathway. They can cross-talk. They can all happen together. They can only happen either one of them. So that's the complication of signal transduction pathway. Okay. Any one of them can interfere with the other one. Okay. So when you are studying signal transduction pathway, if I just wanted to study at ERK pathway, I need to inhibit the other two pathways to make sure the result or the data I see is purely from this pathway. Okay? So that's something you have to keep in mind whenever you're doing your experiment. Then at the end is affecting the transcription factor. One of them is a C gene or C phos. And if you see a C, meaning it's in the cytoplasm, phosphorylation will cause them to translocate it into a nucleus. Okay? And that will drive the cell go into the cell cycle. When you are in the, that's the start side of the cell cycle, when the cell receives the signal. When you, and another checkpoint is the G1S checkpoint. These are the checkpoints you can play with cancer cell. These are the checkpoints you should think of. If that happens in a cancer cell, can I use them as a target for treatment? Right? Okay, so the second one is G1S checkpoint. And the major mechanism here is the RB and E2F. Do you know what is E2F? The factor for transcription. This is the transcription factor. Okay, we start the transcription. RB is a tumor suppressor gene. Okay, and before we know it's the tumor suppressor gene. Well, in here we already know it's a tumor suppressor gene, and then scientists come back to study why it's tumor suppressor gene. We'll talk about RB tumor suppressor gene later, but the function of RB is binding to E2F. By binding to E2F, the E2F cannot do the transcription. Okay, so the kinase come in the interface. The kinase is CDC, cyclin D and CDK 4 and 6, right? Phosphorylate who? Who is the target? Phosphorylate the RB. The RB change the structure and will no longer binding to E2F. The E2F will be binding to gene that is required for S phase. Okay, so that's the regulation, normal cell cycle regulation during the transition from G1 to F. So if this is a cancer cell, the E2F is binding to the gene all the time, right? If I try to kill the cancer cell, these are the targets I could use for the treatment, right? Think about it, because that's your homework. That's part of your homework. So that's RB will inhibit E2F. There are many family members in RB. Some of them is 130 kilodalton size. The acetylation will also inhibit the E2F at the same time. So in order to activate the transcription, you need phosphorylation from CDK4, right? Or CDK2, whatever. The CDK4 or CDK2 is controlled by CKI, the inhibitor for the CDK. One of them is P16, it's very famous, or it's called INK4A. It's an inhibitor for CDK4. So in the presence of your P16, no transcription, because no kinase activity, no phosphorylation, therefore no transcription. Got it? In the presence of P21, okay? No kinase activity. RB is on E2F, therefore no transcription. Got it? Okay. So here's our P53. That's another tumor suppressor gene. And we'll talk about it later. So P53 will activate these inhibitors to inhibit the kinase, to inhibit the cell cycle. Got it? So now everything is in one big picture, right? Somebody has to control this P53. And the molecule here is called ARF. ARF is ADP ribosylation factor that will inhibit P53. P53, we will talk about it later. It's a sensor for a lot of cell activity, including DNA mutation, apoptosis? No.
No?
No.
I thought they guard the gene from mutating. Like, let's
So
say.
we'll talk about P53 later. Okay? So that's the diagram. After we know about cell cycle, this RBE2F will cause gene 1 arrest. Phosphorylation will freeze it, and then you can have transcription. The CKI inhibit the kinase, will inhibit the transcription. The P53 will inhibit, will also inhibit the kinase. Okay, so in the normal cell, when the DNA is damaged, okay, and then you have high level of wild type P53, you will get G1 arrest. You will arrest right here. The P53 is acting on all those inhibitor and kinase. Okay, that's the normal cell. If you have a cancer cell, if you have a mutated P53, or you don't have enough P53, nobody is giving the break. We will talk about how P53 is sensing the DNA damage. Okay. By arresting at G1 phase, allows cell to do the repairing, gene repairing. Okay. That's the main purpose. That would be it today. Let me tell you about your homework. So, your homework, you have to ask any of the AI website, or you can use your, you can ask ChatGPT, you can ask your Gemini, whatever you like. And then I want you to design a novel compound for cancer treatment, okay? By looking at the cell cycle. So you have to choose one checkpoint that is defined in your cell cycle of a cancer cell. If you know a cancer that has this defect in cell cycle, fine. If you don't know, you don't have to write down the name for the cancer cell. Okay? The second step is try to describe the regulatory mechanism. When it is in a normal cell cycle, what does this checkpoint do? So what happens when it's in a cancer cell? Okay? So these two, when you write your homework, you have to write down the mechanism and write down the checkpoint. The third one, ask your AI, based on the mechanism, based on the checkpoint, can ChatGPT design a compound that does the work? Okay? Just ask it. And you have 2 weeks to do this homework. Don't write too many pages, okay? I don't have picture. More than 1 page is enough.
1 page.
Yeah, no more than 2.
Okay.
Okay, no more than 2. You can have 2 pages. I'm fine with it, okay? I wanted to know your logic and how you ask the AI to help your research.
Yeah.
Yeah. And if, for the number 3 step, please write down the prompt.
Oh,
Yeah.
prompt.
Yeah, prompt.
You want to see our prompt?
Yes.
Okay. How creative we are.
Yes. That's the most important part. And we'll discuss it when 2 weeks later.
You know, I just give the screenshot.
Let me see. You just give me this creature. So on the, please turning the homework on the 14th, and we will discuss it on the 21st. Okay, we'll discuss everyone's answer on the 21st when we were talking about the treatment. That will be your treatment. Okay? So next week we'll be here 1 o'clock, and on the 14th please come in on the 12:30. Okay? That will be it for today. See you next week. Bye.